So welcome back to those of you who've been here before. I do recognize quite a few of those names. So welcome to those of you who have never been here before. We are actually the Marine Environmental Education Center. We are a small facility located out on Hollywood Beach, Florida, and we are affiliated with Nova Southeastern University and Broward County. Unfortunately, since we are a small facility, we are still closed. It's not quite possible for us to socially distance at this time. So we wanted to keep engaging with the public. We wanted to be able to provide some cool resources for people to learn about different marine science topics, environmental science topics. So we started this webinar series. We are very lucky because of our affiliation with NOVA and because of everyone that works here, just knowing people in the field. We've reached out to all of our awesome science friends, and they are kind enough to donate their time to the MEEC to provide these really interesting webinars. For those of you who have been here, you know we are very turtly focused. We have Captain the Green Sea Turtle on site. So we'd love to sort of center the day around her. She loves the attention, but we are getting an awesome opportunity right now to sort of expand our field of expertise. Today, we are lucky enough to be talking to Estrella Malka. She is a larval fish ecologist working in CIMAS with UM. 2 years ago, she decided to further her academic career by becoming a PhD student with Dr. Tracy Sutton at Nova Southeastern University. So today she's going to be teaching us a little bit about larval fish ecology. We are going to keep everyone muted just like we usually do, so you can most clearly hear Estrella. So if you do have questions, comments, concerns, if you have any sort of technical issues, please put them into that chat. We will be monitoring that throughout the presentation. And if you have any specific questions, throw them in the chat and we will get to them at the end. We are going to have a couple questions for you as well, so make sure you're paying attention, stay on your toes. And if you want to answer those questions, there is a little raise hand button at the bottom of your screen. If you click that, I will let you unmute yourself. I just need to give you permission, and then you could unmute and participate with the day, okay? I think that's about it for me. So whenever you're ready, feel free to take it away.
Hey, hello. Thank you, Taylor. My name is Estrella Malka, and I'm a PhD student at Nova Southeastern University, as Taylor mentioned. But I've actually been working as a larval fish ecologist at a larval fish lab in the NOAA Southeast Fisheries Science Center, which is a federal building located in Virginia Key, which for those of you who are familiar with South Florida, it's right around near Key Biscayne, really close to, the city of Miami and downtown. So we're at a little bit of a small campus. There's 3 marine institutions in our little island. So we're at the University of Miami's marine campus is located there. My institution, which is the NOAA building, and there's another NOAA lab as well that studies atmospheric science right across the street. So before COVID, of course, we all used to, go to each other's talks and have a very vibrant community. So it's really great to, share a little bit of our research and practice talking to other humans. And so thank you so much for joining me, and hopefully you'll learn something about larval fish. And for those of you who don't really understand what that means, so larval fish ecology, if you see the images here, you have a baby fish. So this is a tuna, a baby bluefin tuna. And then on the other extreme, you have an adult snapper. And he looks like he was just on his way, and a diver maybe took a photo of him, and here he is. And then in between here, you have a very complicated diagram, which is a model of what the ecosystem looks like. And then we have a ship. This is a research ship parked out. I think it looks like it's in St. Thomas in the Virgin Islands. So these are some of my tools that I use for my work, the ships, the fish, of course. And what do? We make models to explain what happens to the adults. So in one slide, this is a little bit of what I do, but more fun is the next slide, which is where I want to ask you if you can guess what fish are you. So let's see, I'm gonna maybe start with an easy question. So I hope you can see my cursor, and I'm gonna try to move it slowly. So just to orient you, we have 1, 2, 3, 4 rows, right? Top row over here. So I'm gonna go to the top right, So can somebody guess? So this is a baby fish. Can somebody guess what that is? And maybe Taylor can help me with that.
Yeah, if you want to give it a shot, just do the little raise hand button at the bottom or get the attention in the chat. We do have a couple people writing in the chat if you want me to shout those out.
I only have one small screen, so please, Taylor, help.
Yep, Morgan and Michelle both say it is a baby seahorse.
Very good. That's great. So that was an easy one, just to make sure you were paying attention and you know how to use the, the buttons and whatever. That was a Zoom quiz. All right. So you passed that one. So what about this guy over here? So this one's a tough one. Just give me a guess. Any fish you think it is? You may have seen him in a sandwich menu.
We have Lee saying it is an eel.
This guy, well, that's a very good guess, but actually that is a mahi-mahi or dolphin fish. It's not a dolphin, it's a mahi-mahi. It's just to make sure. Some people, when they say dolphin fish, they get very upset because they think we're eating dolphins, but that's not, no. Dolphin food meat would probably very oily and not taste very good. But no, so this is a mahi, which is something you might have seen in a sandwich at a restaurant, especially in Florida. They're all, mahi, that's catch of the day often, and they're, they're fast growing fish. So you can see here that they have right here on the top, this guy's very young, but you can see he's got a lot of fins or, happening going on over here. All right, so let's see, what about, somebody said eel, so I'll give you this one. So this is actually an eel, and he is very, very flat. So the eels, like the eels you might see in a reef or in an aquarium, they're very, very similar to bonefish and other fish like that. So they have this larval stage called a Leptocephalus stage, which is like a spaghetti looking stage. So they're pretty much transparent. This particular guy is cool. He's got this like cool spots that go along. And if you kind of look closely, you can kind of see through the other side of the fish. So this spot actually is on the other side, but because they're so transparent, you can see right through it. So and this is their stomach. So this is their very, very small head, which when they're adults, if anybody remembers what an eel looks like, they look very, very, very different. All right, so let's see, let's do one more. Maybe I'll tell you one and you can tell me what you think. So there are two flounders. If you can maybe type in the chat if you like which row, or you can raise your hand. So there's two flounders, different species, different types.
We already have Michelle saying the one, there's river bottom of the very, bottom of the very right.
That's very good guess. Exactly. So he is a flatfish. So that's flounder. So they're completely laterally compressed. So as larvae, actually, this guy is only has one eye right here. And what happens is the eye migrates to the other side. And it happens during the larval stage. So when they're actually born, they're born like normal fish. They have one eye on either side of their heads. But then one migrates through, and I've actually seen specimens where you kind of see it in the process, and it looks really crazy to see, the eye kind of migrating through. But then eventually they both have them right here, and then they will complete their larval stage and their juvenile stage with the two eyes on the same side as most flounders do. So, all right. So I know I don't have all day and we could probably play this game. But so let's see, let's give you the answers. And then maybe you can learn, maybe I'll go over one. So this is a snapper right here. This is the third row on the left. So like a lot of the yellowtail snappers or gray snappers, if you've ever been to a mangrove or near like a dock, you often see them swimming around very, very quickly. They have very strong muscles and they can swim really, really well. And let's see, maybe we can do this guy over here. He's pretty strange looking. So he's a grouper. And a grouper is the family is called serranids. And they're very diverse family. So you have fish that you can eat and fish that kind of more hang out, and they're pretty small. So they're very diverse in size. You have Goliath groupers, for example, blackfin groupers, and there's some other guys that are sea basses, and they are related because of a lot of different taxonomists have looked at their bones and looked at their structure and maybe looked at their genes and said, okay, you guys are, related, and figured out that they're in one family. So, and this is something that's of course been, ichthyology is the study of fish, and it's been, it's one of the oldest sciences for in the marine environment. So anyway, if you have questions, we can always go back to check out this screen, but I wanted to share a little bit more of my research with you, and we can play the larval fish game anytime you want. Okay, so what is my goal is to contribute to the ecosystem. So how do I do that? I use larval fish ecology as my tool to answer questions. So what does that really mean? It sounds very complicated. So let me give you an example. So for instance, this is the life cycle of the Atlantic bluefin tuna. This is a delicious fish that is often seen as a very expensive item on a sushi menu. It's also been hunted or been fished since ancient Roman times. It was seen as a delicacy. So this fish has been targeted for thousands of years by humans, right? They have very valuable price in the market today. So it's also a very commercially valuable fish. So this is right here in the magnifying glass this is the stage that I focus on, right? I focus on the early life history or the larval stage of these fish. So, but to take a moment to talk about the rest of the life history. So how do I contribute to the ecosystem? The ecosystem includes the fish, the ocean, its habitat, but it also includes humans. Now, of course, these fish have existed before we evolved into humans, but, we're here and we're sharing the planet. So, now we are part, of the ecosystem. Of course, this is a very human way to look at the world, but I'm human, so it's okay. So how do we humans manage the ecosystem? So this is an example. So this is the adult fish right here. And once they're old enough, they're mature, we can fish them and, make money by selling them if you are a commercial fisherman. So this map here of the world represents, these arrows represent where the fish go. So this tuna is a highly migratory species, which means it migrates a lot. So it can move, all these arrows is basically where the fish can go. So that's a long distance. So how do you as a human, agency, government, or institution, how can you manage this species? So it's really difficult. It can be very expensive. But one of the ways that we can do that is by focusing on the early life history of the fish. So for example, for this particular species, because I mentioned they've been fished for a long time, their numbers have declined quite a bit. So their stocks have not been doing very well, meaning that we've consumed too many and there's a lot less than there used to be before humans started targeting them. So we can tell a lot by looking at the early life history of particularly populations like this species that don't have a lot of numbers. So however, when you get to other species, it becomes a little bit more complicated. So it's important to pick a species or a family that you can answer questions with. So for bluefin, they have eggs like that are about 1 millimeter. So they're really tiny. They're like the size of the lead of your pencil, basically a little bit smaller than that. But then when they hatch out, they're about 3 millimeters, which is really like about a grain of rice or less, actually. So really tiny. And then by a year, they are, about this big, about the size of your pencil. And they can, swim actually quite a lot. So they're an interesting species. It's the main species that I focus in right now, but let's look at the general life cycle of fish. So larvae have this strategy called pelagic larval stage, which pelagic means open ocean. So that means that they're floating in the ocean, and then they can disperse. So dispersal means that they can move, right? And if you're a very, very tiny grain of rice floating in a very, very big ocean, this might be your only shot to move and your only opportunity to see something else other than your, where you were born. So that's why the larval phase is very, very important. So we were talking about tunas that, move all over the place. Now let's talk about a parrotfish. So a parrotfish, you've probably seen them in aquariums, or if you've been diving or scuba diving, snorkeling, they're very colorful typically. They have the big chomping mouth and they eat the coral. They're very, very important to maintain the health of our coral reefs. You may have heard about that in other webinars or in school, or if you're, work in the field, you may understand what that a little bit more, but, they're really important for the ecosystem. They're not particularly targeted for their commercial value in this part of the world, but in other parts, like I'll mention in a little bit, they actually are fished and they are considered commercially important. But they are important ecologically, which means, we need to study them and understand a little bit more about them. So as adults, they're benthic, which means they're bottom associated. And some of them are pelagic, which means they are, open ocean. But for the most part, parrotfish are benthic. So then you've got some juveniles, and as you can see, the juveniles and the larvae, they don't really look a lot like the adults, right? Here's a larval parrotfish. It's very flat. It's not very colorful. It doesn't have very strong muscles yet. It has this weird hole here, but that's just its stomach. It's just the way that it looks. As they grow older, they're gonna, metamorphose into this beautiful, colorful baby fish, and then eventually become an adult parrotfish. But so all larval fish have this larval stage. Not all of them are pelagic or, open ocean, but the majority of fish are. So it's an important stage. And so in this journey, say, these fish can encounter a wide range of environment and oceanographic variables like, warmer water, colder water. As I mentioned, this might be an opportunity for them to move. So why is that important? So for example, if you have some disease that comes to your reef and you're all very, very closely related, you may not be able to survive that disease and you might be wiped out by that. So that's why it's really important to have genetic diversity in the gene pool of all species. All right, so what is step one if I want to do some larval fish research? So as I mentioned, I use larval fish as a tool to study the ecosystem, but first you need to develop your research question. So the days of basic research are not among us anymore, and we all have to have an applied question, like why do you want to know what, So you can't just I just want to study larval fish. Now you have to have a more specific question. Like, for example, I want to know what the spawning grounds are. Spawning means that's where the reproduction happens. So I want to learn where, for example, tuna reproduce. Okay, that's a good question. Timing, when does that happen? Does it happen every month? January, February, March, April? Does it only happen in the summer? Is it, does it happen during the day, during the night? So there's whole fields of research that look at that in adults. And there's a lot of very cool techniques that you can use to study that. There's another question called connectivity. So what is that? So the ocean it has a lot of currents that move at different speeds. So a lot of these larval fish are connected. So a lot of the times you hear, oh, what is the connectivity of populations? You may have heard that about sea turtles, right? That they're connected to, different parts of the ocean or different coastlines. So, connectivity of larval fish is also very important. Has a lot of different implications. And then one thing you need to integrate is the all life stages. So you can't just study, one stage and call it a day. You need to focus on all of them. So that's why, early life history or larval fish ecology is important, because it's the early life stages. So we may know something about the adults, but if you don't know where they're spawning or reproducing and how often or how important that is, you don't really know everything about the entire life history of one particular fish, right? So, and it's important to understand baseline. So what is a baseline? So if we know what happened, 50 or 100 years ago, that would be our baseline. So what happens now? So that's a trend, right? We want to see patterns and how they change over time, and include how it changes with the seasons and with the years. And then all of this we need to apply to look at fisheries management. And just to show you, oh, here, I can play that again. So this guy over here is, well, he won't play again. He was a larval fish and he was eating for the first time. So this was a paper that some folks did where they were interested to see what are the mechanics. So for those of you that are interested in engineering and other life sciences or physics, and you might say, well, who cares about larval fish? Well, and actually by the way their mouth opens, it creates a vacuum. And that's one of the ways that they're able to eat, because they open it and then they suck water in, and then they're able to let's see, you can maybe, if we go back, we can, yeah, there he goes. So there he goes. He went in there. And then, but you can also look at his body mechanics. And those of you interested in mechanics, that's also really important. You can see how he sort of flexes all of his energy and his muscles to be able to carry out this very, very crucial task, right? It's not just about doing your homework for him, like he needs to feed to survive. So, and the bottom picture here looks like an X-ray, right? Like maybe I colored it with a colored pencil or a crayon. It's actually me taking a sneak peek at what the bones of these fish look like. So these are larval fish. They're only a few millimeters old. My colleague Aki cleared and stained them. So put some chemicals in to highlight their colors, and we can tell things about them. So continuing with step one, we need to think about our management needs. So what is that management? I mentioned it in the previous slide. Fisheries management, that is an effective reason for you to be doing your research today. So how many fish can we remove? That's one question you can have fisheries management answer. But also you need to think about, I'm sure you've heard about, climate change impacts and how our ocean is warming, and different parts of the world are responding a little bit differently. So, but if we don't know what's going on now, how can we predict the future? So for example, changes with temperature, with ocean currents, with chemistry of as it relates to the dispersal and development of these fishes. What are their tolerances and how is that changing? So for example, here in Hollywood, Florida, maybe you're starting to see fish that you didn't see before because it's warmer, or for example, further north in Maine, if you go up the East Coast, you'll see fish that you didn't see before. So those are some of the differences that we're starting to see with a change. And we want to understand, what are these baselines, as I mentioned before. So another thing that we cannot ignore, as I mentioned, is the human and social responses. So you and me, we are human, and we have an impact on our ecosystem and on the fish. Whether or not you've never eaten a fish in your entire life, you're still impacting the ecosystem and the ocean. So these guys over here can't be ignored, because you want to, let's say, preserve the ocean and you don't want to eat fish, and that's absolutely fine, and that's a great goal. But these guys are working, this is their livelihood, and you also have to take their needs into consideration when we're managing the ecosystem. We can't just think about the fish or think about the humans. We kind of have to think of everybody together. So let's see. So how do we do this? So let's pretend we're out sampling. So I'm on a research vessel, right? Typically when I do this, but I want to understand the fish. But first I need to understand the environment. And one of the tools that I use to do that is right here on the left. It's called a CTD, which is an instrument that is, you see this right here, it is towed, it is very heavy, it has these two huge lead weights, and as you can see, it is tied down with some heavy rope on the deck of the ship. So this instrument has these gray bottles, and these bottles hold water. And what happens is that if you look over here on the right, these two students right here, they are recovering it. So what happens is this big crane on the ship is lowered, and it lowers the package, as we call it, down to various depths. It can go as, shallow as 5 meters, and, there are CTD casts that go to thousands of meters, which take forever. But, they're a way for us to see what happens under the ocean, because we can only see the very, very top, right? And as soon as you can't see anymore, I don't know if you've ever dropped something over the water, even in a fountain, right? You throw a coin, often you can see it, but if you throw that same coin in a mangrove, you probably can't see it anymore. So what's going on under there. So this is one of the tools that helps us understand what's going on deep. So let me show you what that means. So in this image right here, you've got temperature and depth. So this is, well, we live in the South Florida, so this is more typical of our area. If you lived further north, this is maybe what you would experience. So this is 25 degrees, which is room temperature, where you don't need a sweater, you're just very comfortable, right? And it's a little bit warmer. So then what happens, as soon as we put the package in the water, we start measuring temperature, and it drops a lot. So it goes from 20, above 25 degrees, which is, very comfortable, to, 5 degrees, which means, you need, if you live in Florida, you need a lot to keep warm if you're used to our temperatures. So this instrument right here can be loaded with tons of things that tells you what's going on underneath the ocean, the very surface of the ocean. You can measure temperature, salinity, oxygen, and that can tell you a lot of different things depending on what your question is. So over here is a picture of one of our interns, Lucy. This was a couple years ago, and Dr. Karen Self, and she's teaching Lucy, who's doing an internship in our lab, how to do this from the lab. So you basically have to communicate with the bridge. You have to communicate with the winch operator, so the people driving the ship, the people driving the cranes, and the people back there, to make sure everybody is safe and that you're collecting the best data that you can. And then you do this maybe 100 or 200 times, and you're done, right? Okay, so that's step 2, sample the environment. And then where I'm more involved in is plankton sampling. So how do you do that? First, you need a plankton net. So this is a bongo net. So a bongo, I don't know if you've ever seen a bongo. It's like, a musical instrument. So that's why it's called a bongo net, because it's got 2, just like a bongo. And then this is an image of a GoPro that we put on the net itself right here, so we could see, what is the fish eye's perspective. And this is this thing here that you see is spinning. It's called a flow meter. And what happens is that it's measuring the amount of water that's being towed. So right now the ship is driving, right? It's going, it's towing, and then now so you really can't see fish going in here, but there's tiny little things that sort of appear now and then. And that's plankton basically, no? So what do we do after we do our tow? We rinse. And rinse very carefully, because these organisms are very, very fragile. They're used to being in the ocean, but we want to study them and see what's going on with them. So we carefully rinse them, and then we repeat that, about 100 times or so, and then you get your plankton. So this is a typical plankton jar, and you can see there's just stuff, right? You don't really know what's in it. So this is through a microscope. And I don't know if you can kind of make this guy out right here. You see there's a fish eye, there's a mouth, and that's a fish. So that's a barracuda, actually. Tiny little barracuda. He's about the size of my fingernail, maybe a little bit longer because they're a little bit bigger. So this was probably a coastal sample, because barracudas are a little bit more coastal. So, but in this particular instance, we were targeting tuna. So this is a bunch of tunas, and if you see, they have big eyes, they have a big mouth, which is a little bit different than some of the fish that we saw in that beginning slide. But as larvae, they're very, very tiny. So these guys are about the size of a grain of rice, very, very tiny. And you can see that we have different sizes. We have got some that are bigger, some that are tiny. And anyway, so once you got the species that you want, you have to identify it, right? So this is another very interesting looking fish. So this one is a larval fish, and it's got this very interesting spines that go up on the dorsal, right up their head. It's a very crazy looking fish, right? So what could this guy possibly be? So he's actually, he's not necessarily a Goliath grouper, but he's in that family of fish. So that's, it's really, really challenging to identify some of these fish. But some of the tools that we use is, for example, we have these drawings, or they're called illustrations, where they highlight differences and similarities. So for example, these are some images that were drawn of parrotfish. And we saw a picture of what an adult parrotfish looked like a few slides ago, right? So these are the larvae. And you may say, well, this is black and white. I can't see anything. Like, why? Why can't I just look at a picture? This is so much prettier. Show me a photo. Well, what happens with parrotfish is that they don't really have much color. They're pretty much black and white, and especially after we catch them. So it's really hard to see. So I want to point you to two spots. So do you see this spot right here, right by its head? It's not in here, right? It's blank here. And that's basically a big thing. So somebody spent their whole, career trying to figure that out, and they did. So that's one way that you can figure out the differences. And that might seem very trivial and unimportant, and say, well, they're just parrotfish, right? But, you can't really say that to dog people, right? Dog people know there are different types of dogs. And it's very, very important to have, whether it's a Labrador or a poodle or, a Chihuahua or, I don't know a lot of dog types, but so it is very, very important. And especially for species, because if they don't, if you don't know what species you are, then how can you figure out everything else? You need to know who they are. So, what is that fish basically? So these are some of the tools that we use. And then once you figure out who everybody is, which, is quite a lot of work actually. So let me take you through some examples. So for example, you've got these fish over here. These are, let's say, reef fish, right? So they live as adults. They like to hang out by the reefs. They're more bottom associated. And you've got labrads and parrotfish, snappers, and groupers, right? So this is some images of those guys. They have, as adults, they like the reef, they like the bottom. And then some of these pelagic fish, and I talked to you about pelagic or open ocean, right? So more like this habitat. That's the mahi, the dolphin fish. So that's a different image of a dolphin fish. And these are some tunas. So they are pelagic fish, which means they're out in the open ocean and they spend their entire life out in the open ocean. So they never find a reef home. That's just not their thing. And I wanted to share with you a couple of examples of larval fish as research tools in the real world. In my case, two projects that we've been working on. The first one is in the Virgin Islands. So the U.S. Virgin Islands Reef Study was funded by the Coral Reef Conservation Program. So as I mentioned, you need a research question first, right? What is my research question? And then you need to plan it. But before that, you need to get money, right? You need to have funding to do this. So you need to find funding either through a grant, which means you need to have a question and apply for this grant, or there's a government funding, which can be federal, like NOAA, for example, the National Oceanic and Atmospheric Administration. has a lot of grants, but there's a lot of other institutions like the National Science Foundation. There's local governments, like for example, there's Fish and Wildlife. There's other local institutions like Waterkeeper, for example. They do a lot of different work in different areas depending on the question. So, but typically for any project that is, a little bit larger, it needs to be a multi-agency collaborative project. And then other fancy words you hear are like interdisciplinary, multidisciplinary, which just means that people that are doing different things contribute differently to the project, right? But so everybody has to collaborate, get along. Typically, for example, in these projects, we've been working with 2 government agencies within NOAA, which is the Southeast Fisheries Science Center, and the Atlantic Oceanographic Meteorological Laboratory, which is the lab across the street. We have also Fish and Wildlife out in the Virgin Islands, and then we have different universities. Now, this is a map of the Virgin Islands, but of the Eastern Caribbean. So this is Puerto Rico here on the left, and this is St. Thomas, St. John, the British Virgin Islands, beautiful areas. This is St. Croix, which is another US Virgin Island. And then you've got this big spans of ocean. And then if you keep going this way, you'll hit Europe eventually, or Africa, depending where you go. Let's just kind of orient yourself. And then the different colors kind of show you the different islands. But I want to point your attention to the yellow. So the bottom here is that a phrase you probably heard before called marine protected areas. So marine protected areas are one of the tools that managers, ecosystem, fisheries, ecologists, governments use to protect different areas. And certain marine protected areas have different goals in mind. So one of them is right here, this little triangle. It's a little bit hard to see, sorry, but it's an important area because you see the different in color here. So this is depth. So this is shallow, right? And then suddenly this starts to drop off and be very, very deep. So this is a map that includes the depth. So it's called, has a map that has bathymetry or depth. And so in this particular area, you can tell that it goes from light blue to white. And then there's all of the structure, right? So there's something really special happening here, which I'll show you in a little bit more detail. But this is where a lot of fish go and hang out and when they want to reproduce. So it seems to be like a really important area. And we were interested, that was our question. We wanted to understand, what is happening with respect to the larval fish ecology of this region. But we ended up sampling this whole area eventually as our project grew. So these are our tracks. So I mentioned that we use a research vessel. So we go and we sample. So we put, points in a map, and then we go do our CTD, which was the water, right? Measuring the environment. And then we do our plankton tows, which is catching the fish. So we did that a lot for several years. So we did that we did it in 2 iterations from 2007 to 2011. And then we went again and were able to sample more recently to see if there were any changes. So, and luckily during every survey, we're able to take students. We've actually taken a high school student, actually she's right here, Adrian, students of PhD, masters, other scientists, government folks, and a couple of volunteers sometimes, and are able to carry this out and teach them what we're doing. So if you see the different colors, is because we did slightly different things. It doesn't really matter, but the point is that we sampled a lot. So once, and let's see, right here. So that area that we were looking at is right here in this area, the important reproductive area, right? So let's just show you what we got. So once you collect that many fish and data, like you need to be able to display it, like, okay, well, show me what you got. That's a big challenge for scientists. One way to do it is to split the area into regions. So for example, I mentioned this is the Virgin Islands, right? So north, south, for example, you can split it in that area. You can split it into east and west. So that's one way to do it. You can do it by island. You have to see what makes sense, you know, and you can take hints at what other people have done in the past, other researchers. So that's called a literature search, right? You want to see what other people have done and how your research can complement their work as well. So let's see. My favorite island is this island because it looks like a drumstick. But anyway, that's just my own observation. So this very exciting graphic is some of our larval fish results. So this is what it looks like after, you know, hundreds and hundreds of these stations. But I just want to draw your attention to the green. So let's pick the groupers because we talked about those, the guy with the big spines and the Goliath grouper. So that family, it's called serranids. So if we look at that over time, as I mentioned, we want to look at trends over time. So what do we see? So we see that these guys are always within the top 75% of abundance. So in this little box, you have the most abundant taxa or the most abundant families that are represented by our toes, right? So, and then you want to see what are the similarities, what are the differences over time. And that was our goal for that particular project, is to look at the overall distribution of the different fish families in the area, because that had not been done before ever. So this is a close-up of that area that I mentioned, the yellow marine protected area. So what could be so special about this area? Well, if you look here, here's St. Thomas and St. John, these beautiful islands. This is really shallow. It's called the Puerto Rican shelf, and then it drops off significantly. So right here is like a huge valley, sort of like the Grand Canyon, let's say. So this particular area, this structure, it seems to be is what attracting these adult fish that spawn. So that's what we were looking at in this particular question. And then there's another bank here, which is called Grammatic Bank. So these are seasonal closures. So that during the time of reproduction, they're protected. They're protecting the adults so that the fishermen don't go out there and eat them all, right? And this is really, really important so that the fishermen can have a job for the rest of the year, right? And not only them, but their kids and so on and so forth. So that's part of management, that you need to manage your resources accordingly. So I mentioned the horizontal distribution, and the vertical distribution is, you know, from the surface to the bottom. So most larval fish, though, you only find them in the top 100 meters. So that's not very deep. But this area can be very, very deep. So the area here, so remember the blue shallow area? So I removed the bathymetry or depth just to make it less confusing. In this particular plot, you've got depths from 15 meters, which is very, very shallow, to 4,000 meters. So that's really, really, really deep, right? So that's kind of this area right here, right in the middle of the trench, let's say. Not quite as deep as a trench, but it's pretty deep. So what are the differences? So we had talked about those reef fish and those pelagic fish, those two groups a few slides ago. So what are their differences over this area of bathymetry? So that's, for example, what you can do now that you know where they are, right? So what we find is for the wrasses, there really was no association. They were everywhere in about normal numbers. But for parrotfish, which, you know, hang out at the reef, interestingly, they higher densities were found in deeper water, which is really, really a unique finding for this particular fish, because, you know, they hang out more in shallow benthic areas. So it must be something dealing with their life history that they have to have, you know, this part of their life there. Otherwise, it wouldn't happen. So for the snapper, though, we see them in more shallow areas. So this is depth here. So this is the 0 to 4,000 meters, which is the deepest that this area goes. But you don't really see that for the fish that are pelagic, which are the blue right here. You don't really see an association because it doesn't really matter for them. Oh, and this is a parrotfish. Remember I told you that they're pretty much black and white. That's exactly what they look like. Another example of some of the work that we can do, which is, you know, way more specific, is you don't really know who they are. So you can use a couple of techniques. You can use some statistics to group them together, and that's what this is, using cluster analysis to see how similar they are, but you're using genetics. So you can tell who they are by how their genes group together. So this is one way for you to identify them when you have no idea what they look like. What happens is that they look very, very similar. And this causes a problem because, you know, if you're trying to identify how diverse, how many fish use a particular reef or area, you don't know. And maybe you're just thinking, oh, it's just one grouper, but it's actually 10 different species. So that's why it's important to understand, you know, who lives there. So, and then maybe the last example I'll share with you, because I'm running out of time, of course, is my parrotfish friends, right? So we can see that there's different species of them. So where are they? And this is, for example, an example of what I mentioned about trends over time and how that, you know, can help you figure out what's going on. Well, I just want to show you this particular plot right here on the bottom. And then this is the abundance of parrotfish. So it looks like they have the most abundance here. So if I were a manager in the area, maybe I want to pay attention to this, right? And see, okay, well, maybe I need to protect this the parrotfish in this area and this island because there's a lot of them. And, you know, what is special about that area? So then that, of course, any of our findings typically end up in a lot more questions. So some conclusions for this particular work, you know, that we have a baseline of who lives here. We've done a lot of surveys and we've contributed to a lot of different regional initiatives like modeling, to fishery councils. We've done some, you know, we've published some of our work and presented it and shared it with the public and other scientists as well, of course. So I mentioned that there were going to be 2 examples. So I'll just share you this one. This was very complicated looking. Don't get scared. We're looking at nitrogen and how that flows through the food chain. So I'm not going to talk about that at all. But so this was a survey we did 2 years. And this is in the Gulf of Mexico. And this was a research vessel that we used. And we, again, bongo nets, bongo, and then there's a mini bongo or a baby bongo that we also use to look at what are the fish eating. So as I mentioned, it's trophic dynamic. So we want to know everything about the fish, not just where they are, but also what they're eating and what other, how is the nitrogen flowing through the food chain. So we were looking for bluefin tuna and we found them. We were very lucky. They're not very easy to catch. And then we measured a lot of things. So this is just a couple of our results. And these are like, what are they eating? So they're eating a lot of different zooplankton in different ways. And this is just one way that you can display that. And if you are interested in some more of that, we can talk about it. But you would think that such a small larval fish would not really have a preference, right? They would just eat whatever. But just like you know, you may love chocolate or not like chocolate. It seems like larval fish have preferences as well. And then what I specialize is in otolith work. So I don't know if any of you may have seen a tree ring, but this is very similar to a tree. If you've ever seen a tree that's been cut across, right? You see this kind of banding pattern of dark and light. So that's what an otolith looks like if you look at it in a high magnification microscope. So this is at 1,000 magnification. And actually all bony fish have otoliths right here in their heads. This is a baby bluefin tuna. And this is me looking at one on the ship. So I study their growth. I age them, and then I try to figure out, you know, why does that change and why is that important? But the whole goal, of course, of all of these studies is to figure out what's going on. And this was in the very, very first slide for those of you who have good eyes and we're paying attention, and we're trying to figure out what's going on. So this is a fish right here, bluefin. And this is kind of the output, like how do we get from here all the way here? So it can be very, very complicated. And that's one of the questions we're trying to answer. And we're getting there. And but along the way, we have a lot of undergraduate research students that have participated and have done work with us. We have graduate students, and it's not that they're not important, but they're just their names. PhD and masters. We have publications and a lot more coming. And then back to, all of the fish. And these are all photos taken by my colleague Lulu Vasquez in Ecosur, which is an institution in Mexico, in southern Mexico. And again, more very cool looking fish. So you may say, okay, well, there's a lot of problems in the ocean. Why do we want to focus on these larval fish? Well, I mentioned looking at trends over time. So this guy over here, and maybe Taylor can help me. What do you think this fish is? Anybody? Look at his fins. I know he's black and white and not very, but he's got, very large pectoral fins, which is your pecs are here, and that's the fish. So it's pectoral fins.
We have a guess, a lionfish.
Very good. Congratulations. Okay.
Lovely.
That is a lionfish. Who guessed that?
We had Lee guess that it was a lionfish. Also, Carly guessed it as well.
Okay, good. Well, good eye. So yes, this is a lionfish, and that's why it's really important to understand the community and not just one particular fish, because we want to, the lionfish, I'm sure as a lot of you have heard, is an invasive species that is suddenly now all over the Atlantic Ocean. And they're native to the Pacific reefs, but they have come in probably in several waves, but now they're established, and which means that they are living and reproducing very actively all over the Atlantic Ocean. And it's a problem because they eat a lot of baby fish. And when you have an invasive species like lionfish, the ecosystem did not evolve with lionfish in it. So the ecosystem is having a hard time dealing with that. So luckily for humans, they are delicious, so we can eat them and remove them. But, they are now part of the plankton community. So we have to think about those. And anyway, there's some more cool fish in here. Some of them are more deep water associated, like this guy right here with those crazy fangs. That was from a deeper collection. That is not a reef fish collection. That's more along what my advisor Tracy Sutton studies. This guy too. This guy is completely see-through. You can actually see through him. He's I don't know what the common name is. Gigantidae is the family name. But anyway, as you can see, there's a lot of different cool types, and I could show you pictures of larval fish all day. But, back to why we care is, besides telling us where and when they reproduce, we can tell a lot from larval fish, and I hope that I've shown you a few examples that we can, model things like their habitat, their movement, where they are. We can learn a lot from just individual fish, like by looking at their otoliths or their ear stones to figure out how old they are. We can look at food webs, like who's eating who and how much. And then of course we have to link it to the environment and to the ecosystem so we can manage it and understand it better. And this is a baby tuna. And of course a satellite, because without satellites at sea, we would be very, very confused. Anyway, and of course, my funding mainly is from NOAA and some previously from NASA, some from our colleagues in Spain with the Ecology project, the Coral Reef Conservation Program, and we have a lot of collaborators and a lot of people that are involved. This is not my research by any means. This is a group of people, and we've been working on this for, over a decade. And hopefully we'll continue to work on that. So anyway, I think I've talked long enough.
Very cool. Thank you so much. I definitely learned a ton. As I said at the beginning, I'm not an ichthyologist, and I learned a heck of a lot all about fish. We do have some questions. Would you like me to read them off if you can't access the chat?
Please.
That's fine. Let's see. Scrolling through all the answers from our beginning. What sort of refuge do larval pelagic fish use when they're still so small and fragile? I know a lot of coral / coastal fish can hide in nurseries? Do pelagic fish just hang out?
So pelagic fish do not. So they're out in the mercy of the ocean. So in some instances, as they're a little older, they may hang out with some floating debris. So like sargassum, which is like the floating seaweed or other objects, but typically they're just they're playing the numbers game. So their parents make a lot of them so that, you know, one of them will make it and survive. So for them, you know, you have maybe a brother or a sister, or if you're like me, if you're a lonely child, you know, it's just you out there, and the, you know, the fate of your species is basically on your shoulders. But for them, you know, they've got, you know, millions of brothers and sisters when they start out, and, you know, all it takes is for one of them to make it. So sad story, but 99.99% of fish will die. So for example, from when they are eggs and they're, you know, they're alive, not only, you know, 0.01% will make it. So it's a really tough world. So the next time you see a fish, whether it's in an aquarium or, you know, in a supermarket frozen, or, you need to just, you know, tip your hat off to them, because they made it and they survived, and hopefully they reproduced before they were caught. So.
Yeah, I actually have a follow-up to that. Is there like a significant difference between how quickly they reach the age of maturity, if it's going to be like a coastal fish that could have some refuge, or if it's a pelagic fish?
Yes, actually, the pelagic fish typically have very short, you know, duration. So they have to grow quickly, eat fast, and develop, so they can get out of this very, very vulnerable early life stage, right? Because there's a wall of mouths, as they call it. They're trying to eat. You know, you're like a yummy potato chip or a cookie, if that's your thing, and you know everybody's trying to eat you because you fit. So yes, so definitely the coastal fish have longer durations typically than the pelagic.
Very cool. We have, let's see, do they have deal vertical migration?
Yeah, so you do see that in some species. You don't necessarily always see that. For some of them are a little bit more marked. Like for example, the tunas, they don't really migrate very deep. So it seems like they're getting whatever it is that they need in the top 50 meters. But like lionfish, you do see them up to 100 meters. So reef fish, typically, you'll see them at different depths.
So what do you think is the main driver behind larval fish propagation? Propagating in areas they don't inhabit as adults, like those, the reef fish that were going into greater depths when they were larvae.
Oh, so a lot of the times they just got lost. So, you know, for example, they, even though they can disperse for a very, very long time, more commonly it's only like, you know, tens of kilometers that they disperse, but that's actually, you know, sufficient for them to maintain their population numbers. So they don't really need to go, like, for example, technically, if I were in Belize, I could, you know, end up here in South Florida because of the way the currents are. The currents are like a highway. For those of you who remember Finding Nemo, you know, Nemo uses the currents, the East Atlantic current, to make his way, right? To kind of fast track his way to find his. Oh no, Nemo is the one that's there. Sorry, I got confused. But you know what I mean? It's a current and it's a highway. So the highways are very important, but a lot of the times the fish get lost. So just because we found them there, it doesn't mean that they're populating those reefs. So that's why you need to kind of understand the whole life history. So if I find a fish out in Antarctica, it doesn't mean that suddenly lionfish populated it there. You know, it you know, likely we'll find a lionfish popsicle, but anyway.
Very cool. We do have Lee asking what larval fish is number 15. I think it's towards the center.
15. Sorry, I'm trying to look for it. 16, this one? This guy?
15, the one above it. Yep.
This guy, let me see. I think he's like a blenny, some kind of reef fish. Let's see. I can tell you. I think I do have the key. I can cheat. That guy, Ogilvius. So yeah, I think he's some kind of like blenny, like a reef, like a small reef fish associated. I will have to, I do not have every single fish memorized because they are literally hundreds and thousands of them. So let's see, SP.
Totally understandable.
All right. Sorry, I can try and figure out. Some of these actually don't even have common names because they are so, rare. Oh, it's a Barracuda. It's a cusk eel.
Very cool. Don't know what that looks like as an adult even, but it looks very cool as a larval fish.
It looks a little bit like, okay, maybe not.
We do have someone that it rolls right into this question. Are there many fish species where their larval stage has yet to be identified and only the adult stage is known to science?
Yes, actually, there's one species of lanternfish that they had only known. They thought that it was a whale fish. I think that they thought about that there was only one species, but it actually ends up that there's two different species. What happens is that with genetics we can figure out who they are, but with genetics, as you will know or learn if you're studying marine science, is that you need to know who they are before you can identify them. So if we have adults, we may not know what the larval stage looks like because they've never been found. That's particularly true for deep sea fish because we're still learning a lot about them. So we may know what the adults are, but we don't know what the larvae are, particularly because even though deep sea fish live in the deep sea, like 2,000 meters, 3,000 meters, their larvae are still floating in the top 50 to 100 meters of the ocean during their pelagic larval duration. And then at some point, they will migrate down.
Very cool. I think that's the last of their questions. I have one more question for myself and for everyone here who seems to be avid larval fish ecologists in the making. Are there any skills or advice that you could give to someone who's interested in getting into this field? Like a lot of times we have like get boat certified, get field experience. So what would you recommend?
Yeah, sure. You can send me an email for sure if you are interested in. It is not a very large world, no pun intended. Well, yes, pun intended. And so a lot of us know each other. So it's not a very large field. For example, here at the University of Miami, we used to have several people that were larval fish ecologists. Now there's a lot less of us, and they've moved to different locations. But there's an emerging side of the aquaculture world where they're very interested to understand, what maximizes production. So that's kind of a really interesting, kind of very applicable job, for example, to, if you're into aquariums, the aquaculture world is certainly an interesting application. Well, for me, I went to undergrad as a biologist, but I really liked marine science. So that's what I started to get into. And then I thought I wanted to be a vet, like a veterinary researcher, but then I fell in love with fish. So then that's what I do now. But try different things, try to be exposed to as many different, if you have no idea what you want to do, just try a little bit of everything until something sparks your curiosity and your interest. And it's not easy. Science is not for everyone, and there's different levels of it. Luckily, like you can make a difference in so many different ways. And so you can, just pick up trash. That's a huge, huge help. And, let's say you go for a walk today, pick something up. You can clean up the beach. You can do different things. So all of that is helping our ecosystem, our environment. Try to reduce your footprint, of course. But if you are interested in larval fish research, feel free to shoot me an email. We used to have interns all the time, but of course everything is closed now. But hopefully, if you're interested to see different fish, we've definitely done events where we'll go to the school, bring microscopes, bring fish, and bring the fish to you. So that's if a particular school is interested, we can do that.
Awesome. Well, thank you so much. We did have someone ask where the recording is going to be for this. So this is a recorded session. We are going to do a quick edit and then put it onto our YouTube page. So just find us on YouTube, Facebook, Instagram, Twitter, any of the socials at Seek the Meek, SEK, the MEEC Meek. We will be sure to post it as soon as possible. Otherwise, if you are free, we are finishing up our November series. We're not going to be doing it during Thanksgiving break, but we are going to have one more before we break for the holidays. So next Tuesday, we actually have Dr. Amy C. Hirons coming in. She is also a NOVA affiliate. She is going to be doing marine mammal and trophic ecology. So come and join us then. It is the same link if you guys are free then. Otherwise, thank you all for coming today, and thank you so much. That was an awesome presentation. I definitely learned a lot. I know that everyone here seemed to have a lot of very cool questions as well.
Yeah, for sure. Yes. As Taylor said, feel free to contact me. And, if you're interested in internship opportunities, unfortunately, you typically have to be 18 and over to go to sea. That's a question that I've gotten before. But right now, the world is limited. But if you're younger and patient, maybe we can do something in the future.
Absolutely. Yeah. And if you think of any questions later on, you can email us at MEEC@nova.edu, and we will pass it right along. Otherwise, yeah, thanks guys. Everyone have a good one and stay safe out there.